Induced pluripotent stem cells, or iPSC in short, are a type of stem cell very similar to embryonic stem cells. They have two main attributes. First, they can self-renew, meaning you can expand the cells while keeping them in a pluripotent state. In addition, upon providing appropriate instructions to the cells in the form of, for example, signaling molecules and defined growth conditions, one can also turn these cells into almost any other cell type of the human body, such as adipocytes, blood cells, neurocells, and many others. iPSCs are derived through an ex vivo cell engineering process, which is often referred to as cellular reprogramming. For this, somatic cells obtained from donors or patients, for example, in the form of skin fibroblasts or certain type of blood cells, are provided with a set of defined factors, also known as reprogramming factors, that over the course of a few weeks will transform initially somatic cells back into a pluripotent state. Now, iPSCs hold immense potential for a variety of life science applications, including disease modeling, drug discovery, and tissue regeneration, because they provide an almost unlimited source of cell material to generate very insightful and specific culture models, which would mostly not be possible otherwise. The entire field of iPSC research has been largely pioneered by Professor Shinya Yamanaka and his team, who showed that mature cells can become pluripotent and identified the factors required for cellular reprogramming. Professor Shinya Yamanaka was awarded the Nobel Prize of Physiology or Medicine together with Sir John Burden in 2012, shortly after the landmark publication who showed of how to put back somatic cells into a state of pluripotency. Culturing iPSCs is relatively complex and more cumbersome compared to other cell systems. The cells require coatings to be able to adhere and grow, and they also need frequent exchange of culture media, which is actually mainly due to some media components being unstable and thus needing frequent replenishment. Based on our recent iPSC cell culture survey, which is freely accessible under the link displayed in the lower left, around 50% of survey participants exchange medium on a daily basis. Now, iPSCs are prone to undergo spontaneous differentiation in culture, and therefore it is really important to monitor the cultures closely for differentiated cells. There are really two main morphological quality attributes to look out for here. First, iPSC's colonies should have defined borders throughout, and second, cells should display a high ratio of nucleus to cytoplasm, as for example shown on the left lower image. In contrast, as shown in the image to the right, colonies with less defined edges and areas and with looser cells and a rather fibroblastic appearance, as highlighted within the red square, should really be avoided, as those are clear signs of differentiation. Finally, overall, iPSC cells also tend to be quite sensitive to manual handling and should be treated very gentle to avoid excessive cell death, increased differentiation, and therefore loss of pluripotency. Isolating or cloning single cells is a very common requirement in the iPSC field. There are several applications for which isolating single cells for analysis or cloning single cells is really crucial. Single cell cloning generally aims to establish homogeneous cell populations from an initially heterogeneous culture, and CRISPR-Cas9 based gene editing workflows, for example, is a very prevalent occurrence in the iPSC field. Single cell cloning is critical here to outgrow appropriately edited single cells into homogeneous populations of cells that can then be used for downstream applications such as disease modeling and organoid culture. Besides cloning, isolation and analysis of single cells is highly relevant for various omics types approaches in the iPSC field, such as single cell proteomics and single cell lipidomics, which are already further enhancing functional characterization and understanding of iPSCs and respective disease models. Now, as I mentioned earlier, iPS cells are comparably sensitive cells, and therefore appropriate and gentle techniques are required for isolating intact single cells efficiently to avoid sample loss, or in the case of single cell cloning, to outgrow individual iPS cells successfully and efficiently. When it comes to isolating or handling single cells, there are various approaches available, including FACS, cell dispensers, and of course, manual techniques such as limiting dilution. Now, whereas manual techniques are quite simple to set up, they are tedious and limited in throughput, and they especially lack proof of successful single-cell isolation or monoclonality. On the other hand, FACS is a more hands-off approach with a more complex setup, but is often deemed comparably harsh on cells and therefore can cause compromised viability. Aside from a choice between manual and automated approaches, there are also different culture vessels used for isolating single cells, for example, petri dishes, or different well plate formats such as 96 or 384 well plates. At IOTA Sciences, we offer comprehensive and integrated solutions for single-cell isolation and cloning, including compact liquid handlers that automate single-cell dispensing, feeding, and the extraction of single cells or clonal cultures, as well as an add-on imaging system that can flexibly be added and automates the verification of monoclonality within the culture chamber. The liquid handlers require only nanoliter volumes per chamber for isolating, feeding, and extracting cells, which greatly saves on consumable expenses. And as you can also see in the data on cloning efficiency shown in the right image, our products handle single cells very gentle, as demonstrated by the market-leading cloning efficiencies of between 70 to 90% for induced pluripotent stem cells, as demonstrated in the graph. Now, if you would like to know more about our offers and ultra-low volume culture chambers for single cells, please visit us at iotasciences.com for further information.